Yes po, sir.
Okay, so hopefully this will be our last. So membrane. So when we have certain groups we want translocated, so we chemically modify it so that it is stuck within the cell. No? So that can happen uh, to certain molecules as well. Okay, so for some prokaryotic cells, they may have photosynthetic abilities, no? So they have chlorophylls inside. So these structures consist of infolding of the plasma membrane. So includes chlorophylls or bacteriochlorophylls. So for even for plants, yung membrane structures are called thylakoids. Okay. So in bacteria, the membrane structures are called chromatophores, lamellae, or chlorosomes. Okay, so let's talk about the cell wall. So as mentioned earlier, it's important for the cell to or the bacteria or the archaea to survive conditions. Uh, having a cell wall will protect against the osmotic pressure in the environment. Okay, so the major component of the bacterial cell wall is the peptidoglycan. Okay, so from the word peptide, you have amino acids joined together, and glycan. So you have the word glycogen. You have a carbohydrate component. So each layer of the peptidoglycan is composed of long chains of alternating molecules of NAG, so N-acetyl glucosamine, and then you have N-acetyl uramic acid. So yan yung structure ng peptidoglycan. But the structures of the peptidoglycan is different in gram-positive and gram-negative bacteria. So in gram-positive bacteria, you have an additional pentapeptide. So it's made up of glycine that cross-link into the tetrapeptide. Okay? Whereas dun sa gram-negative, straight lang yung pagka cross-link niya. Okay? So, yan yung difference ng mga peptidoglycan ng sa gram-positive and gram-negative. So, gram-negative, yung tetrapeptide chains are extended from each NAM unit. Yung gram-positive bacteria mo, meron kang pentapeptide chain. So, yung peptide na yan gawa sa glycine. So, yung cross bridge sa tetrapeptide. Okay? So, I've stated already in the biochem, no? Yung peptidoglycan, yan yung target uh, ng penicillin. So, yung production ng peptidoglycan. So, para hindi magkaroon ng cell wall yung bacteria, and that's the reason why penicillin can kill bacteria. Yung antibiotic properties niya. So, antibiotic drugs are designed to interfere with peptidoglycan synthesis, weakening the cell walls, making the bacterial cell walls more susceptible to the effect of osmotic pressure. Okay, so we don't don't have to remember all of this. Pero this diagram shows you the different antibiotics, yung target ng antibiotics. So I would like to emphasize, no, yung penicillin nga, yung target niya yung cell wall, production ng cell wall ng bacteria. So other antibiotics, they target, for example, DNA synthesis. So meron kang may levofloxacin, and then you have moxifloxacin. So yan mga tina-target niya mga DNA synthesis, RNA synthesis mo. Yan tina-target niya yung rhythromycins. Again, these are drugs that are used to fight bacterial infection. Okay? So nakalagay dyan yung mga specific na mga drugs ginagamit to target a specific molecule in the production of each organelle. Okay? So let's look at the cell walls. So the Gram staining protocol is used to differentiate the two common types of cell wall structures. So the gram-positive cells have a cell wall consisting of many layers of peptidoglycan totaling to 30 to 100 nanometers in thickness. Okay? So in addition, yung gram-positive mo may teichoic acid ka. You have carbohydrate chains that extend through and beyond the peptidoglycan layer. In gram-negative cells, so the walls are thinner. So you have thinner layers of peptidoglycan. And in gram-negative cells, you have the periplasmic space. So between the cell wall and the plasma membrane. Tapos in addition, meron ka ring outer membrane. So for gram-negative, besides the cell membrane, which we can call the inner membrane na kasi may outer membrane ka pa sa gram-negative. So, kumbaga, nasa gitna yung cell wall dun sa gram-negative bacteria. So this outer membrane is attached to the peptidoglycan by murine lipoprotein. Okay? So anong content ng outer membrane? So it contains LPS, so lipopolysaccharide. So this is an endotoxin. So ibig sabihin, since naka-attach sa outer membrane ng bacteria, it is a toxin that can lead to infections that is connected to gram-negative bacteria. So it can cause fever and septic shock. So anong component ng LPS molecule? You have the lipid A, you have the A-core polysaccharide, and an O side chain. So tingnan natin yung structure niya. Wala dito. So ito yung structure ng LPS molecule mo. So this is the O antigen. So pag sinamang antigen, our body creates antibodies for this. So you have the core and then the lipid A. So yan yung LPS molecule. So let's go back to the structure, the differences between the cell wall of a gram-positive bacteria and a gram-negative bacteria. So you can see here, mas organized and less complex ang gram-positive, no? So you have teichoic acid that is included in the cell wall of a gram-positive bacteria. So they're structurally simple, and you have a thick layer of peptidoglycan. Compared to gram-negative, mas complex yung structure niya, no? So again, you have the inner membrane, yung plasma membrane natin. Tapos nasa gitna yung periplasmic space. Tapos nandito yung peptidoglycan cell wall. So kita natin ang nipis, no, compared sa gram positive. Kaya mas nag-sustain to ng purple, whereas dito madaling matanggal yung hello, choppy ba ako? Okay naman. Okay pa naman. Hello guys. Okay pa naman ako?
Okay naman po.
Okay. So you have the outer membrane in addition to the inner membrane sa gram negative, no? So you have the O antigen as indicated, may lipopolysaccharide. Tapos meron kang mga uh, pores, no? Yung pwedeng dumaan yung mga molecules. So that's porine. Pero complex nga yung structure niya. This allows molecules to pass through. Okay? So yan, again, yung structure ng gram-negative bacteria. So mas complex yung structure ng cell wall ng gram-negative bacteria. Okay? So sometimes hindi sapat yung gram-positive and gram-negative bacteria. Sometimes you need another type of stain. So the bacteria of the family Mycobacteriaceae have an external layer of waxy mycolic acid. Okay? So mas magagamit mo dyan yung mga acid stain, acid fast stain. So para ma-penetrate yung mycolic acid. So, kasi other staining procedures hindi kaya. Okay? So yung bacteria, pag itong bacteria, it's called as acid fast. Okay? So kita mo na yung for gram-positive bacteria, although gram-positive siya, may waxy mycolic acid. So gagamit ka ng acid fast staining protocol to detect the presence of the cell wall. Okay? So archaeal cell wall, ano ba yung difference niya sa bacterial cell wall? So they do not contain peptidoglycan. So instead, you have a pseudo-peptidoglycan. So instead of NAM, it is replaced with another subunit. No? So they may have a layer of glycoproteins and polysaccharide that serve as the cell wall instead of the pseudo-peptidoglycan. And there are a few na walang cell walls entirely. You also have a glycocalyx. So from the word yung mayroon kang carbohydrate katulad ng glycogen, no? So you have a carbohydrate. So it's a sugar coat of which there are two important types. So you can have capsules or slime layers. Okay? So yung prokaryotic cells may have additional cell envelope. That is the, in addition to the outer membrane or the cell wall, you have additional structures that protect the prokaryotic cell. So ano ba yung capsule? So this is an organized firm layer located outside the cell wall and is usually composed of polysaccharides or proteins. Okay, so additional protection dun sa bacteria aside from the cell wall. And then you have a slime layer. So this is a less organized layer, loosely attached to the cell wall and can be washed off. Kaya nga slime layer. So it can be composed of polysaccharides, glycoproteins, or glycolipids. So glycocalyces allow cells to adhere to surfaces, aiding in the formation of biofilms. So yung mga biofilms na yan, nag-attach sa surface yan. So there are colonies of microbes that are deep. So they form in layers in surfaces and they absorb water like sponge and prevents the loss of water, no? So, yun, hindi yan na-dry out. It also protects cells from predation and hinder the actions of antibiotics and disinfectants. Okay? So, ayan, yung capsules ng so type siya ng glycocalyx. So, pinaprotektahan yan. Kita yan sa microscope. Yung labas, outside the violet stain, yan yung capsule. So the ability to produce a capsule can contribute to a microbe's pathogenicity. So pag may additional layer ka, mas mahirap patayin. Okay? So that increases the ability to cause disease because it makes more difficult for our phagocytic cell to engulf and kill the microorganism. So example yung Streptococcus pneumonia. So this is a microorganism that produces a capsule well known to aid this bacterium's pathogenicity. So yung slime layer nga, another type of cell envelope structure. So this is composed of structural proteins and glycoproteins. So it's found outside the cell wall. Sa archaea, it serves as the cell wall mismo in some archaea. So marami pang studies ng mga scientists dun sa function ng S layer. Kulang pa ng studies dyan. Okay, let's talk about filamentous appendages. Okay, so yung nasa labas nung appendages dun sa cell wall. So many bacterial cells have protein appendages embedded within their cell envelopes that extend outward, allowing interaction with the environment. Okay? So may mga different purposes yan. So they can attach to other surfaces. They provide movement, which we call yung motility, and it can also transfer genetic material. Okay, so you have Frimbry. So this refers to the short bristle-like proteins projecting from the cell's surface by the hundreds. So ang main purpose niya, mag-attach sa surface and to other cells. So this is important in colonization, no? So paggawa ng mga biofilms. Yung pilay mo, this refers to the longer and less numerous appendages. So this helps in attachment to surfaces and also para sa transfer ng DNA. So yung bacteria, nagta-transfer ng genetic material, we call that conjugation. So yung F. pilus or sex pilus, so this is important in that transfer of genetic material. So it only occurs between members of the same generation when two cells physically transfer or exchange parts of their respective genomes. Okay, so yan example ng pili. Okay, next is flagella. So ito important sa movement. So flagella are structures used by cells to move in aqueous environments. Okay? So gawa siya sa protein flagellin and they extend outwards from the cell and spin solution. So anong components ng flagella? So you have a basal body na naka-embed dun sa plasma membrane. Tapos may hook region ka, tapos doon nakakabit sa filament. Okay, so tingnan natin ang difference ng gram-positive bacteria sa gram-negative bacteria. Okay, so sa gram-positive bacteria, tingnan natin na lang yung nasa present sa gram-negative. Sa gram-negative, meron kang L-ring and P-ring na wala sa gram-positive bacteria. Okay? Ano pang wala? Since wala kang outer membrane sa gram-positive bacteria, no? So, dito sa gram-negative, may outer membrane ka pa. So, dito nakakabit yung hook. Okay, hello? Rinig pa ako? So, pareho naman silang may C-ring at saka MS ring. So, kita nyo yung mga difference dun sa cell structure ng gram-positive. Dito sa gram-negative, may additional plasmic space at saka may outer membrane ka. So, dun pa nakakabit yung hook at saka yung filament. Okay, so you have different arrangements of flagella, no? So pag isa lang yung flagella mo, o flagellum mo, so that's monotrichous. Pag sa both ends, meron. So kahit isa lang or tag-isa lang sa both ends, so tawag dyan ay amphitrichous. Pero kung marami ka sa isang end ng cell, marami kang mga flagella, you call that lophotrichous. Pero kung yung entire cell mo covered with flagella, you call that peritrichous. Okay? So yan. So monotrichous, isa lang yung flagellum. Pag dalawa, amphitrichous. Or pwede ring marami yan, no? So amphitrichous, basta both ends. Pag lophotrichous, only one end pero marami siya. Peritrichous, all over the cell wall, cell body. So these bacteria can move in response to external factors or external signals. So for example, for photosynthetic bacteria or archaea, they move towards the light. So that is phototaxis. And they can also be moving towards a magnetic field or aligning to a magnetic field kasi meron siyang inclusions called magnetosomes. So that's called magnetotaxis. You may also move according to the presence of needed chemicals. So moving towards or moving away from harmful chemicals. So magre-repel siya. So yan yung chemotaxis. So, paano nagmo-move yung mga bacteria or mga archaea, no? Pag may mga flagella. So, pag move sila towards a certain target, yung nagra-run ang tawag natin doon. So, it's running Runs ang tawag doon. Tapos pag medyo nag-change siya ng direction, nagde-decrease ng movement, we call that tumble. Okay? So pag nag-rotate yung flagella niya, counterclockwise direction, so tandaan niyo yung counterclockwise. No? So yung kung movement niya, yung left thumb mo, counterclockwise yung pag-spin ng kamay mo. Okay? So yung counterclockwise. So this allows the bacteria to move forward. So pag nagtatumble, so yung tumbling, kumakalat yung mga flagella niya. So it will be rotating in a clockwise direction. So may looping motion, iikot yung body niya. So it prevents meaningful forward movement at nag-re-reorient yung cell towards the direction of the attractant kung ano niyan, needed yung chemical. Okay? So ito yung runs, ito yung tinatawag na runs, no? So moving towards. So pag nagtumble, medyo nag-change yung direction niya. So nag-spread out yung flagella niya, pumipihit. Okay? So ito yung normal movement ng bacteria or archaea kung walang chemical gradient. So paikot-ikot lang siya. Pero pag may presence ng chemical gradient, mas maraming runs, no? So mas maraming movement towards the chemical compared to yung tumbles. So the overall movement towards the higher concentration of the attractant is called chemotaxis. Okay, ito na eukaryotic na tayo. So again, yung mga nasa domain archaea and bacteria, yun lang yung prokaryotic. So the rest are eukaryotic like protozoans, algae, fungi, plants, and animals. Okay, again, presence of the nucleus and membrane-bound organelles. So presence ng endoplasmic reticulum, mitochondria, Golgi apparatus, lysosomes, peroxisomes. So they are held in place by the cytoskeleton. So itong cytoskeleton, ito ay internal network that supports the transport of intracellular components and help maintains the cell shape. Okay, so yan yung eukaryotic cell. Pero since you study this well sa anatomy, we won't concentrate on that. More on prokaryotic cell tayo. So ito na lang yung difference natin pag-aralan. So this will be the last slide that we will have. Okay. So tingnan natin ang difference niya. So unang-una, mas malaki ang eukaryotic cell kaysa prokaryotic cell. Okay. So 20 micrometers ang usually ang eukaryotic cell. So 5 to 20 micrometers. While yung bacteria or archaea, 0.5 to 1 micrometers. Yung surface area to volume ratio niya, since maliit nga yung prokaryotes, high yung surface area to volume ratio, while sa eukaryotes mababa lang. Genome characteristics. So pag sa eukaryotes, marami kang chromosomes. So it can be haploid or diploid. So yung number of chromosomes pwede 23 or 46 sa humans. Okay, it is linear yung DNA niya. And it contains histones. So para yung DNA mo, maging compact mo, kailangan mo ng histones. Ngayon for a bacteria, you have a single chromosome lang, tapos circular yung DNA. And you have haploid lang, haploid chromosomes, and it lacks histones. Dito sa archaea, so single chromosome then circular and haploid, but in archaea, it contains histones, no? So mas malapit yung archaea actually sa evolution dun sa eukaryotes. In terms of cell division, prokaryotes undergo binary fission. Dun sa eukaryotes, you undergo mitosis, yung normal cell division. Tapos pag meiosis, so ano yan? So magdi-divide mga yung sex cells. Membrane lipid composition. So remember, for bacteria and for eukaryotes, they're both ester linked. So straight chain fatty acids for both. Tapos bilayer yan. Okay? So again, yung hydrophilic mo nasa labas, yung hydrophobic mo nasa loob. Ang addition lang sa eukaryotic cells, meron kang sterols. So may cholesterol ka dun sa ano mo, sa cell membrane mo. Ang archaea mo, ito yung iba. So ether link. So instead of ester, ether link. And you have branch isoprenoids. You have bilayers or monolayer ng phospholipids. Cell wall composition. So, peptidoglycan yun sa bacteria or may species na wala. Pag archaea, may pseudo-peptidoglycan ka. Some species glycopeptide, others are gawa lang sa S layer, so mga polysaccharides or proteins. While sa eukaryotes, so pag yung plant cells or algae, cellulose yan. So pwede kang sa mga mollusk or fungi, chitin. Some algae have silica like yung mga diatoms. Most others lack cell walls like animal cells. Motility. So let's talk about motility. You have rigid spiral flagella composed of flagellin sa bacteria. Ganun din sa archaea. Dito sa eukaryotes, pwede kang, for unicellular, meron kang flagella at saka cilia for yung mga mga protozoans, no? Composed of microtubules. Siyempre, yung mga complex animals, wala namang flagella yung cells. Membrane-bound organelles, wala sa prokaryotes, pero meron sa eukaryotes. Endomembrane system, wala sa prokaryotes, pero meron sa eukaryotes. So endoplasmic reticulum, Golgi apparatus, and lysosomes. Ribosomes, so 70S sa prokaryotes. Yung eukaryotes, 80S yung sa cytoplasm, but since may mitochondria siya, 70S. Okay? So I will end here. So that ends our class for today. So I'll just post this recording sa YouTube.